Hi, my name is Zhibo. I'm a postdoc research fellow at National Institutes of Health.
Hi, my name is Chen, and I'm a senior investigator and the chief of the Laboratory of Molecular Imaging and Nanomedicine, National Institutes of Health. Today, we would like to share with you our recent work published in Science Advances on a new class of boron-derived amino acids, also called Boramino acids, for imaging the expression of amino acid transporters, or AATs. This work was funded by the NIBIB Intramural Research Program and was in collaboration with researchers at the National Heart, Lung, and Blood Institute of the NIH, the University of Southern California, and the Xiaoyuan University.
Positron emission tomography, PET, is a cutting-edge imaging technique that produces 3D images of functional processes in the body. A PET scan only uses very small amount of radio tracer, but can provide high imaging contrast from the diseased tissues to the healthy tissues. The most commonly used radio tracer is called FDG, fluorodeoxyglucose. However, 18-fluoride FDG highlights any tissues of high energy consumption, such as inflammation and brain. Therefore, it is not tumor-specific.
Finding a compound that will distinguish between cancer and inflammation, and perhaps even cancers that do not have high glycolysis, would be of high significance in molecular imaging. AATs are ubiquitously expressed in the body and serve as channels to pump nutrients against concentration gradients into cells. AATs are upregulated in many types of cancer and provide nutrients essential for the survival and the proliferation of cancer cells. There appears to be a direct relationship between the transportation of amino acids and the cancer cell replication. Therefore, PET tracers that can accurately and non-invasively assess the expression of AATs in patients would be highly desirable for clinical diagnosis, patient management, and evaluation of anti-cancer drugs.
In this work, we reinvent amino acids through replacing the carboxylate group by the allosteric trifluoroborate. The substituted molecule is known to be free of in vivo metabolism and can be readily 18-fluoride labeled by recently established 18-fluoride, 19-fluoride isotopic exchange technology. The substitution generates an entirely new type of chemicals denoted as Boramino acids. Some representative Boramino acids have been synthesized and tested by computational modeling, cellular uptake assays, and in vivo biological evaluations. As expected, 18-fluoride Boramino acid inhibits high AAT-mediated intracellular uptake. In addition, 18-fluoride Boramino acid has distinctly high uptake in the tumors, as well as rapid clearance from the normal tissues. Notably, the uptake of 18-fluoride Boramino acid in inflammatory regions is almost negligible, suggesting a unique advantage over 18-fluoride FDG, which is currently the gold standard PET tracer for clinical diagnosis.
In summary, we've introduced a new class of amino acid transporter substrates, Boramino acids, which are beyond the naturally occurring amino acids and are characterized by their strong AAT specificity and the versatility in mimicking various amino acids. Boramino acids can be labeled easily with fluorine 18, and they should find wide application in the development of new PET imaging probes for clinical diagnosis, as well as in fluorine 18 labeling of AAT-mediated pharmaceutical candidates, in order to accelerate the evaluation of their biodistribution. Translation of some key AAT substrates into the clinic for cancer imaging is currently ongoing.